Hello, my name is Stephanie Wairagu. I am a senior at Meredith College, majoring in public health with minors in data science, chemistry, and biology. And today I'm happy to share with you my data science research project that is using nonparametric statistical methods to analyze cotton data. So the aim of my research was to investigate the effect of a plant-based biostimulant on cotton, which is one of the agriculturally important crops in North Carolina. I did this by analyzing cotton data that was provided by Matthew Starr of the Department of Crop and Soil Sciences at NC State. Another goal was to create reproducible code. And this was important because this is an ongoing research project looking at the effects of biostimulants on many different crops and not just cotton. However, some of the challenges that may be encountered when dealing with agricultural data include small sample sizes and non-normality of the outcomes. Hence, the focus of my presentation today will be discussing alternative statistical methods that can be used when you analyze data of this manner. Plant biostimulants generally support plant growth and development, and by definition, they are any substance or microorganism that are applied to plants with the aim to enhance nutrition efficiency, abiotic stress tolerance, and or crop quality traits, regardless of the nutrient content of the biostimulant. In this study, the biostimulant we are looking at is called IQ-FORT, and it is produced by a process known as enzymatic hydrolysis, which results in releasing its active materials, which are oligopeptides and free amino acids that are used by the plants. It is also important to note that biostimulants are increasing in popularity, not only because of their ability to increase plant development and productivity, but also because they reduce the need for fertilizers, thus preserving the environment. As we know, sometimes using too much fertilizer can alter the fertility of the soil. And another major concern is that fertilizers contain substances like nitrogen, methane, and carbon dioxide, and the emission of these has contributed to the large quantities of greenhouse gases that are present in our environment. So step one of this data analysis was cleaning up the data. What you see on the left is the raw data that I was given. And as you can see, this format is not ideal for data analysis. Therefore, I cleaned it up, ensuring that every row represented a unique observation. I also removed the columns that were not relevant, and I was only left with the variables that were of interest. Some of the variables that we are testing include the following. So we have lint weight, seed weight, open boll weight, number of open bolls per plant, length, strength, micronair, and uniformity index. Step two was assessing the distributions of these variables using histograms, since most of these were continuous variables. And here is a snapshot of some of them. So the IQ-FORT treated plants are displayed in blue, and the control plants are represented in red. So we have the IQ treated plants in blue and the control plants in red. And as you can see, the distributions for the IQ-FORT treated plants are quite right skewed. So for the purpose of this short presentation, I will focus in on one response variable, and that is lint weight. And as you can see again, the IQ group is very right skewed. Additionally, our sample size was only 20, meaning that we cannot assume normality based on the central limit theorem. This is because for the central limit theorem to work, we need a sample size of 30 crops or more. So to fix the issue of non-normality, I first tried doing a log transformation, which is quite popular when you have right skewed data. And when I did this, I noticed that the IQ treatment group was fixed, but when I looked at the control group, it started to look a bit left skewed after doing the log transformation. So I decided to do a Shapiro-Wilk test to test for normality. And when you're doing Shapiro-Wilk tests, the null hypothesis is usually that the data are normally distributed. And as you can see on the right, when I did the test, I got a p-value of 0.05617 for the control log group, which technically is above the alpha level, meaning that we can reject the null and conclude that the data was normal. However, I was not comfortable with how the distribution looked left skewed when I plotted the histogram, which brings us to another challenge when we're dealing with small sample sizes. So tests for normality, like the Shapiro-Wilk test, are subject to type 2 errors when we have small sample sizes. So for example, in this case, we see that because of the low power, we fail to reject the null hypothesis when in fact the control group is not normally distributed. Therefore, it is always prudent to counter-check the results of the Shapiro-Wilk test, either using a histogram or a normal probability plot, which is shown on the right. So as you can see, the normal probability plot again shows us that the log of the control group is definitely left skewed because we see that the black dots, which are the observations on this lower end, are very far away from the straight line. So what's the solution for all of this? I resorted to using a nonparametric method known as the Wilcoxon rank sum test, which is an alternative for the two-sample t-test that checks for differences in means. So the Wilcoxon rank sum test is based on ranks, which means that it is therefore resistant to outliers, making it suitable when you have data that's not normal. The null hypothesis for Wilcoxon rank sum test is that the two groups that are being compared come from the same population, and the alternative hypothesis is that the two groups being compared come from different populations. It is important to note, however, that since I was testing for multiple hypotheses simultaneously, there was the need to adjust my alpha level of significance using a Bonferroni correction. The main responses that were of importance to our principal investigator were lint weight and seed weight, which are the most important when you're dealing with cotton. So when I did my Bonferroni adjustments, I found that my threshold was now 0.025. And that's what I will compare my p-values with. So here are some results from the Wilcoxon rank sum test. As you can see, both p-values are less than 0.025, meaning that we can reject the null hypothesis at the 0.025 level of significance and conclude that the distributions of the treatment plants and the control plants were significantly different, meaning that the biostimulant did have an effect on the cotton. So my recommendation when dealing with data that has a small sample size and outcomes that are not normally distributed is using non-parametric tests like the Wilcoxon rank sum test, as they're flexible and quite easy to do. Lastly, I wanted to make sure that my code was reproducible. So to do this, I wrote a script with functions that could easily be reused for future crops. And my first function that you can see was to check the distribution of the variables. Then I had another function where I used a Shapiro-Wilk test to check for normality. After that, I plotted some normal probability plots to counter-check the results of the Shapiro-Wilk test. And finally, based on the results of my Shapiro test, I ran a test that checked whether the biostimulant had an effect on the crops. So if my Shapiro test confirmed that the data was normally distributed, my function would run a normal t-test. And if the Shapiro-Wilk test confirmed that the data was not normally distributed, then the function would run a Wilcoxon rank sum test. So in conclusion, again, we can confidently say that nonparametric tests are a good alternative for agricultural data with small sample sizes and non-normal distributions. Our data analysis also revealed that the biostimulant IQ-Fort did have a significant effect on our cotton responses. So as for my future considerations, I will explore using bootstrapping as another alternative approach to nonparametric methods. And for my Bonferroni correction, I am curious to see if another method such as the Holmes procedure is more powerful with a lower probability of type 2 error. So thank you for listening. And I would like to thank my faculty advisor, Dr. Emily Lada, for her guidance throughout this research project. Thank you.